All right, so get ready because today we're diving deep into biochemistry. Looks like someone's been wrestling with some pretty tough exam questions from a B10 biochemistry course.
Yeah, it's definitely a course that really makes you think about those complex processes. But hey, that's why we're here. We're going to untangle some of those tricky concepts and make sure you feel good about this material.
Exactly. And I think you'll be surprised by some of the amazing details we uncover along the way. So we'll be tackling each question one by one, breaking down those answer choices, and making sure you really understand what's going on. Ready to dive in?
Absolutely. Let's go.
Perfect. So let's start with this question about sequencing by ligation and phosphatases.
Ah, yes. This one really gets at the heart of how important it is to, reduce background noise and ensure accuracy in sequencing.
Right. So when we talk about sequencing by ligation, this is a technique that's used to determine the order of nucleotides in a DNA molecule. But this question is really zooming in on the role of phosphatases in that process.
That's right. And phosphatases are the key players here. They're enzymes that remove those 5-phosphate groups from the primers that are used in sequencing by ligation.
Okay, so why is removing those phosphate groups so important? Like, what's that actually doing in terms of making things more accurate?
Well, you can think of it like, you're putting together a really complicated puzzle, but some of the pieces have these extra bits sticking out, and it makes it harder to see the correct fit.
Okay, I can see that.
So these extra phosphate groups, they're kind of like those extra bits. They can lead to what we call unwanted or incomplete ligation products, and that creates all sorts of confusing signals.
So it's like those phosphate groups are adding extra noise to the data, and
Exactly.
the phosphatases come in and kind of clean things up.
Exactly. It's a really subtle but powerful way to increase the accuracy of those sequencing results.
That makes a lot of sense. All right, well, let's move on to another one of these kind of membrane mysteries and talk about lipid bilayers. This question digs deep into the composition and dynamics of these structures, which are, as you know, absolutely crucial for life.
Yeah, absolutely. And what's interesting here is that this question is emphasizing the asymmetric distribution of phospholipids within that lipid bilayer. It's not just a uniform sea of lipids. There's a very specific arrangement, and that's super important for the membrane to function properly.
So if we picture a lipid bilayer like a sandwich, we're talking about different fillings on each side of that sandwich, right?
Exactly.
So what kind of phospholipids are we finding on the outer layer of this cellular sandwich?
So on the outer layer, you're going to find a higher concentration of things like phosphatidylcholine, sphingomyelin, and glycolipids. And these are important for, the stability of the membrane and how it interacts with the environment outside of the cell.
Okay, so that's the outer layer. And what about the inner layer, the one facing the cytoplasm? What's going on there?
Well, the inner layer, it's got a totally different set of characters. You're going to see more phosphatidylserine, phosphatidylinositol, and phosphatidylethanolamine.
Wow. It's amazing how much complexity there is in this seemingly simple structure. So we've got this bilayer of lipids, but the composition of each side is very specific.
Yeah, and that's really important for things like cell signaling and, even processes like apoptosis, which is programmed cell death.
Right. And speaking of complexity, the question also mentions membrane rafts. Can you remind us what those are and why they're important?
Yeah, so membrane rafts, they're like these specialized microdomains within the lipid bilayer. You could think of them almost as little platforms, and they're enriched with specific lipids like cholesterol and sphingolipids.
Okay.
And these rafts, they act as platforms for various cellular processes,
Uh,
things like signal transduction and protein trafficking.
So they're like little clubs within the membrane where specific molecules hang out together to do important jobs.
Exactly. And, the question mentions a technique for studying these rafts, and it suggests electrostatic forces, but that's not really how we visualize them. The best way to see these tiny structures is with something called atomic force microscopy, or AFM.
Ah, AFM. That's a technique that lets researchers visualize the surface of the membrane at a really high resolution so you can actually see those lipid rafts.
Exactly. It's much more powerful than just looking at electrostatic interactions. It gives you a really detailed picture of these structures.
That's incredible. Now back to those phospholipids. The question mentions one particular phospholipid as being an eat me signal. Is that right?
Well, the question specifically mentions phosphocholine, but actually that's not quite accurate. The real eat me signal during apoptosis is phosphatidylserine.
Ah, okay. So when a cell is ready to be removed, it flips this phosphatidylserine from the inner layer to the outer layer, like it's putting up a flag to signal time to go to the immune system.
Exactly. It's basically a beacon that attracts phagocytes, those cells that engulf and remove the dying cell. A very controlled and orderly process.
Fascinating. It's amazing how a simple flip of a phospholipid can trigger such a complex cleanup process.
It is. And speaking of movement, the question also explores how those phospholipids move between the layers of the bilayer.
Yeah.
It's not a static structure at all. There are these three key enzymes involved in this lipid shuffle: flippase, flopase, and scramblase.
So we have flipase flipping phospholipids from the outer to the inner layer, flopase flopping them back out, and scramblase just scrambling them in both directions. That's a lot of activity.
It is, and all that movement is crucial for maintaining that asymmetric distribution of phospholipids, which we know is essential for the cell to function properly.
Right. Okay, so we've talked about the lipid bilayer, but now let's zoom in on another crucial component of those cell membranes proteins. The question specifically asks about type I membrane proteins and their orientation within the membrane.
So type I membrane proteins, they're positioned in a specific way with their C-terminus, that's the end of the protein chain, facing the cytoplasmic side of the membrane.
Oh, okay, so if we picture the protein like a tree, the roots or the C-terminus will be anchored inside the cell and the branches are extending outwards.
Exactly. And that orientation is super important for how the protein interacts with other molecules, both inside and outside the cell.
Makes sense. Now, the question also touches on membrane transporters, specifically symporters and antiporters. These are the proteins that are moving things across the membrane, but they have different modes of operation.
They do. Seaporters, they're all about teamwork. They move two different substances in the same direction, kind of like a carpool lane on the cellular highway.
I love that analogy.
Yeah.
So they're taking advantage of this electrochemical gradient to power that movement.
Yeah. So it's not just about moving things from high concentration to low concentration. There's also this electrical component involved. And symporters, they're really good at using that gradient to their advantage.
So it's like they're riding the wave of this gradient, making it easier to transport things.
Hmm.
What about those antiporters? They're a little different, right?
Yeah, antiporters are more like, they move two substances in opposite directions across that membrane. And they're not just passively riding the gradient, they actually use energy to move things against the concentration gradient.
So they're working against this flow. How do they do that?
Well, they use the energy that's stored in ion gradients, and these gradients, they're created by pumps, like the sodium-potassium pump, for example.
So it's like a revolving door, right? One person's movement is powering the other person's movement. One substance moving down its gradient is providing the energy to move the other substance up its gradient.
Yeah, that's a great way to think about it. And this is really important for the cell to maintain that balance, that homeostasis.
Right. Now, there was also a mention of GPI anchors in the question. Can you refresh our memory on what those are and where they're made?
Ugh. GPI anchors, or glycosylphosphatidylinositol anchors, they're synthesized in the lumen of the endoplasmic reticulum, or the ER. They're basically these lipid carbohydrate structures that attach certain proteins to the cell membrane.
So they're built inside the cell's protein factory, and they end up on the outside of the membrane. What's their purpose once they're attached to a protein?
They basically act as like tethers. They anchor that protein to the outer leaflet of the membrane, and that positioning is really important for how the protein interacts with things outside the cell.
Okay, so we've got these proteins embedded in the membrane. Some are anchored by these GPI anchors. Some span the membrane with specific orientations. It's quite a dynamic picture.
It is.
And we can't forget about, I think, one of the most essential players in this membrane landscape, the sodium-potassium pump. This pump is crucial for maintaining those electrochemical gradients that we talked about earlier. And the question specifically mentioned phosphorylation during the pump's operation.
Right. So the sodium-potassium pump, it's this amazing little machine that uses ATP, the cell's energy currency, to move sodium ions out of the cell and potassium ions in. And it does this against their concentration gradients, which is pretty impressive.
Yeah, it's like it's constantly working to keep things balanced inside and outside the cell.
Exactly. And during this whole transport cycle, the pump itself actually gets phosphorylated.
So it's like the pump gets energized by this phosphorylation event.
You got it. And that phosphorylation causes a change in the pump's shape, which allows it to bind and release those ions on opposite sides of the membrane.
So it's a continuous cycle of phosphorylation and dephosphorylation that keeps the pump going. And that maintains those really important gradients.
Exactly.
Amazing how this tiny pump plays such a fundamental role in cellular life. Now, moving on to another key concept, membrane potentials. This question mentions the Goldman equation.
Yeah. The Goldman equation, it's this really powerful tool because it takes into account the permeability of the membrane to all sorts of different ions, like sodium, potassium, and chloride. So, unlike the Nernst equation, which only looks at a single ion, the Goldman equation considers the contributions of multiple ions. That gives us a much more accurate picture of the overall membrane potential.
Right, so it's a more complete picture of all those charges across the membrane.
Hmm.
The question also asks about why the inner surface of a non-excitable cell is more negative. What's the main ion responsible for that negativity?
Well, the primary culprit is potassium, or K plus size. Even though sodium is more concentrated outside the cell, the membrane is much more permeable to potassium at rest.
So even though there's more sodium outside, it doesn't have as much influence because it can't get through the membrane as easily.
Exactly.
So those potassium ions are flowing out of the cell, and that leaves behind a net negative charge on the inside.
That's right. And that creates what we call the negative resting membrane potential.
Okay, so potassium is the key player. What about chloride ions? What role do they play in all of this?
Chloride ions, they're kind of like the followers. They tend to, go with the flow, so to speak. They'll passively diffuse across the membrane, and that help to balance out the charges and maintain that overall membrane potential that we were talking about.
So it's a carefully orchestrated balance between all these ions, with potassium setting the stage, and then chloride helping to maintain that equilibrium.
Exactly.
Now, before we move on from membranes completely, there's one more question I want to tackle. It's about two different techniques used to analyze proteins, Malte-Tolf and LC-MSMS.
Ah, yes. These are really powerful tools that give us a ton of information about proteins.
Right, so let's start with Malte-Tolf, which stands for Matrix-Assisted Laser Desorption Ionization Time of Flight. Can you break down how that technique works?
Sure. So in MALDI-TOF, you take your protein sample and you embed it in this special matrix. Then you hit it with the laser pulse, and that ionizes the protein molecules and sends them flying down this flight tube.
Okay, and then what?
Well, we measure how long it takes for them to reach a detector at the end of the tube.
So it's like a race, and the lighter ions are going to finish first.
Right. You got it. And from that time, from the time of flight, we can figure out the protein's mass to charge ratio. And in MALDI-TOF, proteins usually only carry a single positive charge.
Got it. One positive charge for Malte-Tolf. Now let's untangle LC-MSMS. That stands for liquid chromatography, mass spectrometry, mass spectrometry. How is that different from Malte-Tolf?
Okay, so LC-MSMS, it's a bit more complex. It involves separating the proteins first, based on their properties, using this technique called liquid chromatography.
So we're sorting the proteins before we analyze them.
Right, and then once they're separated, they go through mass spectrometry.
And what happens in the mass spectrometry stage?
Well, it's similar to MALDI-TOF in that the mass spectrometer is measuring the mass to charge ratio of those protein ions. But with LC-MSMS, we can actually fragment those protein ions and analyze the pieces. And this is where it gets really interesting because those fragments can tell us so much about the protein structure and what it's made of.
Wow, so it's like we're breaking the protein down into clues and then using those clues to figure out its identity.
Exactly. And unlike MALDI-TOF, in LC-MSMS, the proteins can actually carry multiple positive charges, and that gives us even more detailed information.
So it's like a superpowered protein detective.
Exactly. It's incredibly powerful for identifying proteins and for analyzing those post-translational modifications that we were talking about earlier.
Okay, so we've gone deep into membranes. We've talked about cutting-edge protein analysis techniques. I feel like we've only just scratched the surface.
There's so much more to uncover. We haven't even touched on the fascinating world of genetics and gene expression yet.
I know. But don't worry, we're not done yet. We'll be back soon with part 2 of this deep dive, where we'll explore all the intricacies of DNA, genes, and all those amazing molecules that bring them to life.
Definitely. Stay tuned because things are about to get even more interesting. Welcome back to the deep dive. Ready to explore the amazing world of genetics and gene expression.
Absolutely. It's like we've laid the foundation, by talking about the cell's structure, and now we get to zoom in on the blueprints, the DNA, that really dictate everything.
Exactly. And, those blueprints, they're not just static instructions. There are all these incredibly intricate mechanisms that control which genes are expressed, when, and how. That's what makes this whole area so fascinating.
Definitely. Speaking of control mechanisms, there's this one exam question that dives into the lambda-phage life cycle, that virus that infects bacteria.
Right.
And the question is focusing specifically on the roles of those CRO and repressor proteins. Can you remind us how those proteins work together to regulate that switch between the lysogenic and lytic stages?
Sure. So it's almost like this genetic tug of war that's going on. The crow protein, when it binds to this region of the phage DNA called OR2, it pushes the phage toward the elytic cycle. That's the takeover and destroy mode.
So Crow is the aggressive one, ready to unleash chaos on that bacterial cell.
You got it.
What about the repressor protein? That one's trying to, keep things calm, right?
Exactly. The repressor protein, when it binds to the OR1 region, it keeps the phage in its kind of dormant lysogenic state. It's basically silencing the genes that are needed for that elytic cycle.
Okay, so it's this back and forth, this balance between these two proteins that determines what happens to the bacterial cell. Now, the exam question specifically asks about how UV light influences this balance. Does UV light directly switch the phage to the photolytic cycle?
Well, not directly. It's more like it sets off this chain of omics that ends up tipping the scales. So UV light, it damages the bacterial DNA, and that activates a protein called RICA.
RICA, yeah, that rings a bell. That's involved in DNA repair, right?
Exactly. It's a key player in DNA repair. But it also has this other function. It can act as a protease, which means it can break down proteins.
So RICA targets the lambda repressor protein for destruction.
You got it. So it's not the UV light itself that directly flips the switch. It's the DNA damage, and then the RICA activation that removes that repressor, and then the COR2P protein is free to take control and push the phage into that lytic cycle.
Wow. It's amazing how these seemingly unrelated processes, like DNA damage and viral life cycles, can be so interconnected.
Yeah, it's a great example of how intricate and interconnected things are within a cell.
It is. Okay, so let's shift gears to another fascinating example of gene regulation, the lac operon.
The lac operon, a classic example of how bacteria can, turn genes on and off in response to what's going on around them.
Right. Specifically, in this case, the presence of lactose. It's all about efficiency, right?
Yes.
The cell only wants to produce the enzymes needed to break down lactose when lactose is actually available.
Absolutely. Makes sense, right? Why waste energy on something you don't need?
Exactly. Now, the exam question focuses on the molecule that actually triggers this expression of those lactose metabolizing enzymes.
Hmm.
Is it lactose itself?
It's actually, it's close, but not quite. The molecule that does the trick is 1-pyrrole-6-allolactose.
1,6-allolactose, so it's like a slightly modified form of lactose.
Right. And it binds to the like repressor protein. Now, that repressor normally blocks the transcription of those genes that are needed for lactose metabolism, but when 1-pyrrole-6-allolactose binds to it, it changes the repressor's shape and it basically falls off the DNA.
So now RNA polymerase can access the genes and make those enzymes.
Exactly. 1-pyrrole-6-allolactose, it's like the key that unlocks the door to lactose metabolism.
I love that. So we have lactose coming in, it gets modified into this inducer molecule, and boom, the cell starts making the enzymes it needs. But how is 1,6-allolactose even produced?
Well, it's pretty clever, actually. A tiny bit of one of the enzymes involved in lactose breakdown, beta-galactosidase, it's always present in the cell, even when lactose isn't around. So when lactose enters the cell, beta-galactosidase converts a small amount of it into 1-pyrrole-6-allolactose.
So there's always a little bit of that enzyme hanging around, ready to make the inducer when it's needed.
Exactly. It's all about that quick response, being ready to go when lactose is available.
I love how efficient these bacterial systems are. Speaking of efficiency, let's talk about the molecular machinery that actually transcribes those genes, RNA polymerase. There's an exam question that focuses on the prokaryotic version of this enzyme.
Okay.
And it really highlights the importance of that sigma subunit.
Hmm?
Can you remind us what makes the sigma subunit so important for RNA polymerase?
Sure. So the sigma subunit, it's like a guide for the RNA polymerase. It makes sure that the polymerase binds to the correct promoter sequences on the DNA. Those specific sequences that signal the start of a gene.
Right.
Without the sigma factor, the RNA polymerase would just bind randomly to the DNA, and that could lead to the transcription of, all sorts of nonspecific and potentially harmful sequences.
So the sigma subunit brings order to the process,
Yeah.
making sure that the RNA polymerase starts at the right place.
Exactly. Otherwise, it would be chaos. The cell wouldn't be able to control which genes are turned on and off. It would be like having a conductor for an orchestra. Without the conductor, the musicians would just be playing whatever they wanted and there wouldn't be any harmony.
That's a great analogy. So the sigma subunit is the conductor, making sure the genetic orchestra is playing the right tune at the right time.
Exactly.
Now let's dive into another level of control. post-translational modifications. There's an exam question that asks for like 4 examples of these modifications.
Okay.
And it wants us to talk about the specific amino acid residues that they target and what their functions are. I feel like we're getting into the world of protein accessorizing here.
I like that. It's like adding those finishing touches to a protein.
Right. So let's start with phosphorylation. What amino acids are usually targeted by phosphorylation, and how does it change the protein's behavior?
So with phosphorylation, we're adding a phosphate group, and that typically happens to serine, threonine, or tyrosine residues on the protein.
Okay.
And this can do all sorts of things. It can activate the protein, it can deactivate it, it can make it stick to other molecules, it can even mark it for destruction. It's really like flipping a switch on the protein.
Wow, so one little phosphate group can have a huge impact. What about methylation? What's the target there, and what are the effects?
So methylation, it's adding a methyl group, and it often targets lysine or arginine residues. And it's a little more subtle than phosphorylation, but it can definitely have an impact. It can affect gene expression, DNA repair, even protein stability. It's almost like adding a little tag to the protein that changes how it interacts with other molecules.
Okay, so it's a more subtle modification, but still super important for the protein's function.
Yeah.
What about ubiquitination? That one always sounds a bit more ominous to me.
Ubiquitination is unique because it involves attaching a whole other protein called ubiquitin to the target protein, usually at a lysine residue.
Right. We talked about that one earlier. It's like putting a label on the protein that marks it for destruction.
Exactly. It sends the protein to the proteasome, which is like the cell's recycling center.
So it's the ultimate protein cleanup crew. Okay, what about glycosylation? That one sounds a bit sweeter.
Glycosylation, it's all about adding sugar molecules, or glycans, to proteins. And this can affect the protein in a lot of ways. It can affect how it folds, its stability, and even how it interacts with other molecules. It's kind of like decorating a cake. You add those decorations and it changes the whole look and feel of the cake.
I love that. It's not just about making the protein taste better, it's actually changing its structure and function. So we've got phosphorylation, methylation, ubiquitination, and glycosylation, 4 key players in the post-translational modification game, and each one brings something unique to the table.
They do, and they highlight the fact that, protein regulation isn't just about turning genes on and off. It's also about fine-tuning those proteins after they're made, making sure they're doing exactly what they need to do at the right time in the right place.
Exactly. Okay, now let's shift our focus back to that amazing machinery responsible for transcribing DNA into RNA, eukaryotic RNA polymerase II. One exam question asks about the specific factor that phosphorylates its C-terminal domain.
Oh yeah, that's TFIIH. It's a multi-subunit protein complex, and it plays a crucial role in getting transcription started.
Right, TFIIH. It's like the Kickstarter for RNA polymerase II. Can you remind us why this phosphorylation event is so important? What does it actually do?
Well, that phosphorylation, it acts like a signal, basically telling RNA polymerase to release from the promoter and start making that RNA chain. It's like the green light that allows the polymerase to move beyond the starting point and start actually synthesizing the RNA molecule.
So the crucial step in the transition from initiation to elongation, the actual process of making that RNA molecule. Okay, now let's move on to a question about those incredibly useful tools in molecular biology restriction enzymes. These enzymes are like molecular scissors, cutting DNA at very specific sequences, and they're used all the time in labs around the world.
They are.
One of the exam questions asks about how these enzymes are named, using ECRI as an example. Can you break down that name and tell us what each part means?
Sure. So Icari, it comes from Escherichia coli, strain RY13, and it was the first restriction enzyme discovered in this particular strain.
So the name tells us the genus, species, strain, and even the order of discovery.
Exactly. It's like a little passport for the enzyme telling us where it came from and a little bit about its history.
I love that. Okay, let's tackle this next question about mutations. Specifically, it asks about 4 different types of mutations: frameshift, silent, missense, and nonsense. Can you define each type and explain how they might affect the protein that's produced?
Absolutely. So frameshift mutations, they occur when you have nucleotides that are either inserted or deleted from that DNA sequence, and this throws off the reading frame of the genetic code.
Okay, so it's like if you're reading a sentence and suddenly some letters are added or removed, it's going to change the entire meaning of the sentence.
Exactly. And in the same way, a frameshift mutation can change the entire amino acid sequence of the protein, often making it completely non-functional.
So it's like throwing a wrench into the protein synthesis machinery.
Yeah, pretty much.
Now, what about silent mutations? Do they have any effect at all?
Well, silent mutations are interesting because they involve changes in the DNA sequence, but they don't actually change the amino acid that's incorporated into the protein.
How is that possible?
It's because of the redundancy in the genetic code. There are multiple codons that can code for the same amino acid, so you can change the DNA sequence, but if it still codes for the same amino acid, the protein stays the same.
So it's like changing a word in a sentence to a synonym. The meaning stays the same, even though the word is different.
Exactly.
Okay, what about missense mutations?
So missense mutations, they do change the amino acid that's incorporated into the protein.
Okay, so that's going to have an effect.
Yeah, but the effect can vary. It really depends on the specific amino acid change and what role that amino acid plays in the protein's structure and function. Sometimes it can be a minor change, sometimes it can have a really dramatic effect. It's kind of like swapping out one ingredient in a recipe. Sometimes it might make the dish taste a little different, but sometimes it can completely ruin it.
Right. So the impact of a missense mutation really depends on the context. What about nonsense mutations? Those sound pretty, well, pretty final.
Yeah, nonsense mutations, they're the ones that introduce a premature stop codon into the mRNA sequence. So it's like putting a period in the middle of a sentence, the message gets cut short, and the protein synthesis machinery stops prematurely.
So the protein ends up being incomplete.
Exactly. It's truncated, and it's often non-functional.
So we've got frame shift, silent, missense, and nonsense. 4 ways that the genetic code can be altered, and each one can have a different impact on the protein that's produced.
Right. And it's these mutations that are the driving force behind genetic diversity. But they can also lead to diseases and disorders. So understanding how mutations affect protein function is super important, especially if we want to develop treatments and therapies for those diseases.
Absolutely. Now, before we move on to the next question, I wanted to touch on this really interesting concept related to mutations, the wobble effect. It basically explains why some mutations, especially those that affect the third base in a codon, don't always change the amino acid sequence of the protein.
Right. So the Mobile effect, it's all about this flexibility in the base pairing between the third base of a codon on the mRNA and the corresponding anticodon on the tRNA.
Okay, so there's a little bit of wiggle room in that third position.
Exactly. And this flexibility allows for some variation in that third base without changing the amino acid that gets incorporated into the protein.
So that's why we have this redundancy in the genetic code. Multiple codons can code for the same amino acid.
Exactly. And that redundancy, it's like a built-in safety mechanism. Even if there's a mutation in the third base of a codon, there's a good chance it won't affect the protein at all.
That's pretty clever. Okay, now let's switch gears to another fascinating process within the cell. Protein targeting. There's an exam question about how proteins are directed to lysosomes, which are like the cell's recycling centers.
Okay.
What's the signal that tells a protein, hey, you're going to the lysosome?
Well, the signal is a very specific sugar molecule called mannose 6-phosphate, or M6P.
M6P, okay. So proteins that are destined for the lysosome get tagged with this M6P, and that acts like a postal code, telling them where to go.
Exactly. And this tagging happens in the Golgi apparatus, the cell's packaging and distribution center.
Right, so the Golgi recognizes the M6P tag and packages those proteins into vesicles. And those vesicles are then sent specifically to the lysosomes.
You got it. It's like a well-coordinated delivery system within the cell.
I love how organized everything is. Okay, so we've talked about protein targeting. Now let's move on to a question about signal transduction, the process by which cells receive and respond to signals from their environment.
Right. It's like the cell's communication network, allowing it to sense what's going on around it and react accordingly.
Exactly. One exam question focuses on G protein coupled receptors, or GPCRs, which are, really important players in this cellular communication network. The question specifically asks, what happens when a GPCR gets phosphorylated?
Okay, so when a GPCR is activated by a signal, it can be phosphorylated by an enzyme called a kinase. And this phosphorylation creates a binding site for a protein called beta-arrestin.
Okay, so phosphorylation is adding a sort of tag to the GPCR that attracts beta-arrestin.
Right. And when beta-arrestin binds to the receptor, it basically acts like a brake, preventing the GPCR from interacting with the G proteins, which are the next players in the signal transduction pathway.
So it's like turning off the signal.
Exactly.
But what happens to the GPCR after it's bound by beta-arrestin?
Well, the binding of beta-arrestin can also trigger the internalization of the receptor, and that happens through a process called endocytosis.
Endocytosis. That's where the cell basically engulfs something, right? So the GPCR is removed from the cell
You
surface.
got it. And then it can either be recycled back to the surface or it can be targeted for degradation.
So the cell can adjust its sensitivity to the signal by controlling how many receptors are on the surface.
Exactly. It's a very dynamic process, constantly fine-tuning the cell's response.
That's really cool. Now let's move on to another key player in signal transduction, PKA, or protein kinase A. There's a question about the specific amino acid residues that PKA targets.
Oh yeah, PKA is very specific. It only phosphorylates serine and threonine residues, and those amino acids are often part of specific motifs or sequences within the protein, so their phosphorylation can have a really big impact on the protein's activity or where it goes within the cell.
So it's like PKA is a molecular sculptor, adding phosphate groups to very specific sites on the protein and changing its shape and function.
I like that analogy. And PKA is involved in so many different signaling pathways, so that precise targeting is really important.
Definitely. Now let's tackle this question about a specific post-translational modification that can lead to the overproduction of KMP, which is of course a really important signaling molecule.
Okay.
What modification are we talking about here, and how does it affect KMP production?
We're talking about ADP ribosylation, and specifically ADP ribosylation of the G alpha subunit of a G protein. Remember, G proteins are like these molecular switches that transmit signals from those GPCRs that we were talking about.
Right. And the G-alpha subunit can be bound to GDP, which is the off state, or GTP, which is the on state.
Yeah.
And different G-alpha subunits can either activate or inhibit adenyl cyclase, which is the enzyme that makes CAMP.
Exactly. It's all about maintaining that balance, making sure there's just the right amount of CAMP in the cell. But when the G-alpha subunit that activates adenyl cyclase, called GINA, is ADP ribosylated, it gets stuck in its active GTP-bound state.
So the on switch gets stuck, and adenyl cyclase keeps making CAMP.
Exactly. And that overproduction of CAMP can really mess things up in the cell, depending on the specific signaling pathway that's involved.
Wow, it's amazing how one little modification can have such a big impact. Okay, last question for this part of our deep dive. It focuses on two conditions that affect muscle function: myasthenia gravis and botulinum toxin poisoning. Both conditions can lead to muscle weakness, but they do it through different mechanisms. Can you explain the molecular basis for each condition?
Sure. So in myasthenia gravis, what happens is the body produces autoantibodies, and these antibodies target the acetylcholine receptors at the neuromuscular junction.
Okay, so the neuromuscular junction, that's where the nerve meets the muscle, right? And acetylcholine, that's the neurotransmitter that signals the muscle to contract.
Exactly. So when those acetylcholine receptors are blocked by the autoantibodies, that signal from the nerve to the muscle gets disrupted, and that leads to weakness.
So it's like the antibodies are putting up a roadblock, preventing the signal from reaching the muscle.
Yeah, that's a good way to think about it. Now, botulinum toxin, it takes a different approach. It actually prevents the release of acetylcholine from the nerve terminal in the first place.
Oh, wow. So it's like cutting the wires before the signal can even be sent.
Exactly.
Yeah.
It does this by cleaving these proteins called snare proteins, which are essential for the fusion of those synaptic vesicles with the nerve terminal membrane. And those vesicles, they're the ones that contain the acetylcholine.
So if those vesicles can't fuse with the membrane, the acetylcholine can't be released.
Right, and the signal to the muscle never gets sent.
Wow. So both mechanisms disrupt signaling at the neuromuscular junction, but they target different parts of the process.
Exactly. And understanding those different mechanisms is crucial for developing effective treatments for each condition.
That makes a lot of sense. Okay, what an incredible journey we've taken through the world of genetics, gene expression, and signal transduction. We've talked about DNA replication, transcription, translation, all these amazing processes that bring those genes to life. And now we've even touched on how cells communicate with each other.
It really has been a deep dive.
It has. And we still have one more part to this adventure. We'll be back soon to tackle some more fascinating membrane mysteries and protein puzzles.
Definitely. There's still so much more to explore. Welcome back to the deep dive. Ready for the final leg of our biochemistry journey. We've got some more membrane mysteries and protein puzzles to solve.
I'm ready. It feels like we've been on this incredible adventure through the cell, from its DNA blueprints to the intricate communication networks that keep everything running. And now we're back to where we started, that dynamic barrier that defines the cell, the membrane.
It is a fitting end to our exploration. The cell membrane really showcases the elegance and complexity of how cells are organized. And it's amazing how much we can learn from just a few exam questions.
Speaking of those exam questions, one that really stuck with me was about sequencing by ligation. Specifically, the role of phosphatases in ensuring accurate sequencing results. Can you remind us why those enzymes are so crucial?
Absolutely. Imagine you're putting together a really complex DNA puzzle, trying to figure out the order of all those nucleotides. But sometimes those puzzle pieces have these extra bits sticking out. And those extra bits, they can make it hard to get a clear picture of how everything fits together.
So it's like trying to fit puzzle pieces together when some of them have like extra tabs or something. It makes it harder to see the correct connections.
Exactly. And those extra bits, in this case, are 5-phosphate groups on the DNA primers. They can lead to these unwanted or incomplete ligation products, which basically create a lot of noise in the data.
So it's like those phosphate groups are adding extra signals that can make it harder to interpret the results. And phosphatases come in and clean things up.
Exactly. They remove those phosphate groups and reduce that extra signal intensity. That makes it much easier to identify the correct DNA sequence.
Wow, so a seemingly small change can make a big difference in the accuracy of the results. Okay, let's revisit those membrane wraps for a moment. The exam question talked about using electrostatic forces to study them, but if I remember correctly, we talked about a different technique.
You're right. Electrostatic forces, while important for membrane interactions, they're not really specific enough to visualize those tiny membrane rafts. The best way to see these dynamic structures is with atomic force microscopy, or AFM.
Right, AFM, it's like having a superpowered microscope that can zoom in on these tiny membrane neighborhoods and actually show us their composition and how they're organized.
Exactly. AFM allows us to see beyond the average membrane landscape and really appreciate the specialized roles that these rafts play in different cellular
It's
processes.
amazing how much detail we can see with these advanced techniques. Now let's talk about the phospholipids that make up the bulk of the cell membrane. I remember the exam question emphasized that they're not just randomly distributed within the bilayer. There's a specific arrangement, right?
Absolutely. It's not just a homogeneous mix of lipids. The specific arrangement of phospholipids is really important for the membrane to function properly. On the outer layer, the one facing the extracellular environment, you'll find more phosphatidylcholine, sphingomyelin, and glycolipids.
And on the inner layer, facing the cytoplasm, we see more phosphatidylserine, phosphatidylacetal, and phosphatidylazolamine. It's incredible how this seemingly simple structure, a bilayer of lipids, is actually so carefully organized.
It is, and that asymmetry is crucial for things like membrane fluidity, how the membrane interacts with other molecules, and even how it participates in signaling pathways.
The question also mentioned a specific phospholipid that acts as an eat-me signal during apoptosis. Which phospholipid is that, and how does it signal a cell's demise?
The eat me signal is phosphatidylserine. It normally resides on the inner leaflet of the membrane, but during apoptosis, it flips to the outer leaflet.
So it's like the cell is putting up a flag, letting the phagocytes know that it's time to be engulfed and removed.
Exactly. It's a very controlled process that ensures dying cells are removed without causing any harm to surrounding tissues.
Okay, let's shift our focus to those proteins that are embedded within the lipid bilayer. One exam question focused on type I membrane proteins and how they're oriented within the membrane.
Right. So type I membrane proteins, they have their C terminus, that's the end of the protein chain, facing the cytoplasmic side of the membrane.
So if we imagine the protein like a tree, the roots or the C-terminus are anchored inside the cell, while the branches are extending outward into the extracellular space.
Exactly. And that specific orientation is super important for how the protein interacts with other molecules on both sides of the membrane. It influences its role in things like cell signaling, transport, and adhesion.
The exam question also brought up those fascinating membrane transporters, the symporters and antiporters. Can you remind us how those differ in their mode of operation?
Symporters, they're all about teamwork. They move two different substances across the membrane in the same direction, harnessing the energy of the electrochemical gradient to do so.
Like a carpool lane on the cellular highway. What about antiporters? They're a bit more, well, rebellious, right?
That's a good way to put it. Antiporters move two substances in opposite directions, and they actually use energy to move those substances against their concentration gradients.
So it's like one substance going with the flow is powering the movement of another substance against the flow.
Exactly. They use the energy stored in ion gradients, which are created by pumps, like the sodium-potassium pump we talked about earlier.
It's amazing how cells have evolved these intricate mechanisms to move molecules around. Now let's touch on those GPI anchors that we discussed earlier. They attach certain proteins to the cell membrane. Can you remind us where those anchors are synthesized and what their purpose is?
Sure. GPI anchors, or glycosylphosphatidylenyl acetyl anchors, are synthesized in the lumen of the endoplasmic reticulum, or ER. They basically act as tethers, anchoring those proteins to the outer leaflet of the membrane.
So they're ensuring that the protein is positioned correctly to interact with the outside world.
Exactly. And that positioning is often crucial for the protein to carry out its function.
It's another example of the cell's incredible precision in organizing its components. Now, we can't wrap up our membrane discussion without revisiting that amazing little machine, the sodium-potassium pump. It's constantly working to maintain those electrochemical gradients that are so essential for cellular life. And the exam question mentioned phosphorylation as a key part of how the pump works.
That's right. The sodium-potassium pump, it uses ATP to move those sodium ions out of the cell and potassium ions in, and it's doing this against their concentration gradients. So it's like it's constantly swimming upstream.
And during this process, the pump itself gets phosphorylated. It's like it gets a little boost of energy from that phosphate group.
Exactly. That phosphorylation causes the pump to change its shape, which allows it to bind and release those ions on opposite sides of the membrane.
So it's a continuous cycle of phosphorylation and dephosphorylation that keeps the pump going and maintains those essential gradients. It's a truly remarkable machine.
Mm.
Okay, one final question to tackle before we wrap up this deep dive. It's about a crucial concept in cell biology, membrane potentials. And the question specifically mentions the Goldman equation.
The Goldman equation. It's a really powerful tool for understanding membrane potentials because it takes into account the permeability of the membrane to multiple ions, not just a single ion like the Nernst equation.
So it gives us a more accurate picture of the overall charge distribution across the membrane. And speaking of charges, the question also asked why the inner surface of a non-excitable cell is more negative.
Right. And the main ion responsible for that negativity is potassium, or K plus SOBio. Even though sodium is more concentrated outside the cell, potassium is much more permeable at rest.
So potassium is the key player in setting that negative resting membrane potential.
Hmm.
What about chloride ions? What role do they play in maintaining this balance?
Chloride ions, they tend to follow the flow, so to speak. They'll passively diffuse across the membrane, helping to balance out those charges and maintain that overall membrane potential.
So it's like a carefully choreographed dance of ions, with potassium setting the stage and chloride helping to keep everything in equilibrium.
Exactly. And that balance is essential for all sorts of cellular processes, everything from nerve impulses to muscle contractions to nutrient transport.
Wow, what an incredible journey we've taken through the world of biochemistry. We've explored the intricate structure of cell membranes, delved into the fascinating mechanisms of gene expression and signal transduction, and even tackled some challenging exam questions along the way.
It's been a true deep dive. I'm so glad we could explore these fascinating concepts together.
Me too. And to all of you listening out there, thank you for joining us on this biochemical adventure. We hope you've enjoyed exploring the inner workings of cells as much as we have. Until next time, keep those brains buzzing and stay curious.